As Eva mentioned, we'll be talking about our efforts to better understand the autism and ADHD co-occurrence in early development through using clustering or subgrouping approaches. And obviously this work was done in collaboration with Professor Brammer, and he can't be with us today because he's in Australia, but he kindly pre-recorded some slides that I will share with you. So what we know is that about 1 in 5 people are neurodivergent, which can impact their perception, cognition, and experiences. And according to the neurodiversity paradigm, the way an individual processes different types of inputs or stimuli constitute a neurotype. And these neurotypes can be a specific neurodevelopmental condition. So for example, autism, ADHD, intellectual disability. But what we know is that they can often overlap. And it is likely that there can be multiple neurotypes which are not diagnosable per se, so they are not defined by clinicians. And in this webinar, I focus on the autism and ADHD co-occurrence, as epidemiological studies indicate that this overlap is very common and can present in between 40 to 80% of autistic people. And whilst characterizing our sample, I found that over half of preschool children scored above a cutoff, indicative of either a combined form of ADHD or preschool inattentive or hyperactive impulsive type of ADHD, which obviously highlights the high prevalence of this overlap and suggests that we should be better understanding the needs of children who have both conditions. So how does this co-occurrence impact the lives of individuals with both conditions? Well, literature indicates that children who have a diagnosis of both conditions have a poor quality of life, especially health-related quality of life. They also struggle more with functional outcomes. So they have difficulties with adaptive behaviors measured by the Vineland. These children also show an increased likelihood to present with more emotional or behavioral problems, which can then translate to higher incidence of mental health difficulties, such as anxiety or depression. So the question is, what stopped us from studying the precursors of ADHD and autism in early development? Well, research has largely focused on middle childhood, adolescence, and adulthood while studying this overlap. And the reasons why this overlap is vastly understudied are twofold. So we often lacked reliable measures that would tap into specific neurocognitive processes in early development. In preschool years, there were no validated tasks for studying attentional processor executive functions for children who might have varied levels of intellectual disability or might be nonverbal. And this often led to underrepresentation of autistic preschoolers in developmental research. And there were also no large enough cohorts that would allow subgrouping analyses and help us understand early markers of ADHD and autism, as well as identify a child's individual support needs. So during my PhD, I had the pleasure of working on the Preschool Brain Imaging and Behavior Project, otherwise known as PIP, which some of you may be familiar with. So PIP is a multi-site longitudinal study which characterizes neurodivergent and neurotypical preschoolers. We track the development of autistic children as well as children with ADHD, developmental delay, and neurotypical children. And up to this point, we've enrolled over 450 children. We've completed the first time point of the study and are currently finishing the second time point. And at time point 1 and 2, we comprehensively assess children on their behavior, cognition, brain structure, brain function, and genetics, as well as microbiome. And at the third time point, parents fill in questionnaires to provide information about specific outcomes, including things like quality of life. So of importance to this project, in PIP, we also administer a comprehensive task battery that spans across various transdiagnostic neurocognitive domains that were previously implicated in both autism and ADHD. So these tests have been adapted for children with various levels of intellectual disability and are made to be engaging for young populations. So how did we make them engaging for preschoolers? Well, they include gamification, nonverbal demonstrations and adaptations, as well as optimization of speech stimuli through Bayesian optimization. And specifically to better understand ADHD traits and autism, we looked at two biobehavioral domains. So firstly, executive functions, which is an umbrella term for complex cognitive processes and includes things like focusing or problem solving. And in this investigation, I looked at sustained attention, so the ability to focus on a given task, as well as inhibitory control, which is an ability to inhibit prepotent responses, as both of these processes were widely implicated in ADHD. Secondly, we also measure reward processing with a reward learning task, as there are various hypotheses that differences in the reward system are driven by a dysregulated dopaminergic system in ADHD and should be investigated widely. So I want to highlight that I wouldn't be able to conduct my analyses and the results that I'll be reporting later without mixed methodological developments. So I will next play a couple of pre-recorded slides by Mick that explain the clustering pipeline, and then we'll follow with my results.
I want to talk to you today about cluster analysis. We use this quite often in biomedical data analysis. Frequently, we want to identify heterogeneities in data, either for intervention or for understanding. And there are a whole group of methods that we have available, which will extract subgroups of data on various criteria from a large data set. And they differ considerably in what they do. And I'll talk about some of those today and the methods that we've used in the study that Julia is going to talk about. Just to illustrate the issues here, you can see that there are three ways in which you could get two clusters here. I mean, we've got two clusters on the left, which are basically quite tightly bunched groups of data, which are just slightly touching. Then in the middle, we've got completely separated groups of data, which are sort of arc-shaped, completely different shape from the ones on the left. On the right-hand side, we've got a different sort of set of shapes again. In each case, we can say that there are two clusters, but it would be quite difficult to actually try to define clusters that would actually fit all of these three different types of situations. And that's one of the issues. And because of this, there are many different types of data cluster analysis. And that's one of the things that I'm going to talk about today. So possibly one of the most common methods is called the k-means clustering, which basically groups data around centroids. And really, each data point is basically allocated to the nearest centroid. That method actually requires you to say in advance how many groups there are, which is an interesting issue and one I'll come back to later. Another very common method is hierarchical clustering, where we either start with single clusters and aggregate them until we get basically one large cluster, or we start with basically one large cluster and divide it until we get back down to single clusters, and we get something called a clustering tree. And from that, we can actually gain information about clusters. Another method, which is multivariate Gaussian clustering, is if the data are approximately normally distributed, we can use this method. And this is a very attractive method because there's a huge amount of normal theory that we can bring to bear on analyzing data by methods like this. A method which also has achieved some sort of prominence in recent years is spectral clustering, which uses the eigenvalues of a similarity matrix. These are not by any stretch of the imagination all the methods that are available. There are probably literally scores, maybe even hundreds of methods of clustering. All these different methods use different assumptions about the data. For example, the k-means assumes they're approximately globular in shape. Multivariate Gaussian methods obviously assumes there's an underlying Gaussian distribution. Hierarchical clustering assumes things about the way in which the data points are linked to each other and distance measures that you might use. So they all have issues that we need to take into consideration when we use them. So what are true or meaningful clusters? Well, that really is the $64,000 question. And one of the hardest things to do is decide when you've got a reliable clustering result from a set of data. There are many different methods of evaluating the quality of clustering, but one of the major issues is they will frequently differ quite markedly with each other when applied to the same data set. We've actually historically used one method, which is consensus clustering, which is basically to use a lot of different clustering methods and then try and identify clusters which are common across all those methods. we, having looked at that method, we decided there was a bit of a problem with that, in that it would be entirely possible to have a whole range of different methods which agree with each other, but they all made implausible assumptions about the data. So you might get a result that was consistent, but incorrect. So latterly, we've tried to examine the question, is there one method that we can use that will work on all the types of data that we might see in the type of analysis, the type of data sets that we come across? And clearly in the kind of data that Julia is going to talk about today, we frequently have categorical, binary or continuous data. So we really need a method that will work on all of those types of data. And from what I've said previously, you can see that one of the issues is that a lot of methods don't work on all those types of data. Having looked at this for years, we come to the conclusion that possibly the most general way of looking at a cluster is to use density-based clustering. Density-based clustering essentially just assumes that a cluster is a dense, relatively dense collection of data surrounded by areas of lower density. Even then, when we've gone on to use a particular method, we have to make a number of sort of issues about what we're trying to get out of clustering. If we do get differences between different clustering methods, what might that tell us? In a particular clustering that we might get, which variables are driving that cluster? In a particular clustering, again, for an individual, how likely is that individual belongs to a particular cluster rather than another cluster? These are all interesting issues. So our method is based, that I'm going to talk about for the rest of today's presentation, is based on a method called density cut, which was developed in 2016. It's one of the many density-based clustering methods, but it has a number of attractive features which I've developed and exploited to get the method that we've been using in our studies. So the method essentially starts by looking at the nearest neighbours of each point. So basically it simply takes each point, looks at the other points that are closest to that. And it's typical in data, like in approaches like this, to use something called the k nearest neighbours method, where you choose a value of k, and I'll come back to that later. Say k might be 10, so you take the 10 nearest neighbours. And in Ding's original method, the density was simply the reciprocal of the distance to the 10th neighbour. So in other words, the shorter the distance to the 10th neighbour, the higher the density. And you can see that working here. You can see the point on the top left, point I, as a very low density area, and it's quite a long way to the nearest neighbours that we've chosen, whereas points K and J are much more tightly clustered. So in those cases, the density is higher because the distance to the dotted circle, which actually defines the distance to the 10th nearest neighbour in this case, is smaller for K and J than it is for I. So the distance is smaller, so 1 over the distance is larger, and so the density is larger. So there are some issues with using this method of estimating density. Virtually any method of estimating density can be criticised. And in response to that, and this idea didn't come quickly, it took some time to develop this, we decided to use a number of different methods of estimating density, and we chose 3 in the end. One was the original method in the Ding paper. Another method is to use the number of shared nearest neighbours. In other words, how many of the nearest neighbours of a point X are also nearest neighbours of a point Y. The more shared neighbours they have, the more likely it is that this is a high density area. It's just another way of defining density. And it has the property that it works quite well in high dimensional data sets. A third method is called the reverse K nearest neighbours. And basically that's in a particular data set, how many of the points in the distribution have one particular point as a nearest neighbour. So it's turning the sort of K nearest neighbours on its head. By using a combination of those 3 methods, we thought that we might sort of cover all the bases with respect to estimating density. So having got an initial estimate of the density, we can then basically determine the highest density points in the distribution. They're called the modes of the distribution in the Ding paper. And they start off as our sort of cluster centres, if you like. And then we gradually, so we start off with a small number of clusters, And then we can actually gradually make them larger and larger cluster numbers by changing the height of the valley between the clusters relative to the height of the sort of, so if a particular group of two clusters, like on the left-hand side of that picture, you can see that there are two modes and there's a valley between them. By changing the ratio of the valley to the lowest of the two peaks and gradually altering that, we can get a whole hierarchical clustering tree of the data. It's essentially a way of looking at what happens if you perturb the structure more and more and more. How stable are the particular clusters? And that turns out to be a very useful idea because it gives us a way of looking at, to try and find out what the best number of clusters is in the data set. So the final algorithm consists of basically looping over three methods of density estimation, a lot of different k values to define the sort of search space for the nearest neighbours. And also in the Ding paper was this idea that you could take the original density estimate and you could do a sort of random walk on the data sort of to actually refine the density estimate. So we've got 20 levels of refinement of the density estimate, 3 methods of density estimation, and 10 different values of k. So that gives us 600 hierarchical clustering trees, each one of which by shifting the valley to the peak gives you 100 different clusterings. So we end up with 60,000 clusterings. What we then do is simply say we look for the number of clusters that is the most common amongst those 60,000 clusterings, and we take that as being the cluster number in the data set. So this is an example of how this works. This is a very famous test example. It's called the S1 dataset, much used by people who test clustering methods. And it consists of 15 clusters, most of which are approximately globular, but some are not. There's a sort of rather long, thin cluster. You can see there on what we do. We basically see there's a very clear peak at 15 clusters, which in fact is the true number of clusters in the data set. And that's the clustering that we get out of that. So we basically, by choosing the maximum frequency cluster across all of these different cluster estimates, we can get quite a good estimate of the number of clusters in the data set. And this works remarkably well across many different types of data and across many different numbers of clusters, and it does not require pre-specification of clusters. In fact, the thing about this method is it doesn't require pre-specification of anything. We basically, all we do is put the data in and the solution comes out without us specifying neighborhood size or cluster number or whatever. Now that was a good example and a straightforward example, and here is a less clear example where we've got, as you can see, two sets of clusters which are touching, and then 3 others which are completely separate from those two. Now if we run the algorithm on that, we get 7 clusters as a solution. You can see that looks like a perfectly good, plausible solution. But when we look at the frequencies of the clustering solutions, there's a peak at 7 and there's also a peak at 5 clusters. And in fact, that's quite interesting because the 5 cluster solution looks like that. So basically you can see that all we've done there is join together the 2 clusters at the top right into one cluster with a sort of narrow neck connecting it. And the same with the sort of olive green cluster sort of coming across from the left. So this raises the interesting point of what is the correct cluster number. The correct cluster here, there are essentially 2 very similar clusterings. And for that reason, we output the top 3 clusterings from our data on the understanding that people might want to look at more than one solution if there are a lot of solutions of similar frequency. So the original method that Ding elucidated performs well, but it still requires you to put in k and it still requires you to put in alpha, and it uses a single density estimation method. We've actually, by using all 3, we've been able to cluster some of the sort of international test clustering data sets, which Ding's method doesn't cluster very well. The SNN method in particular works very well for large diffuse clusters, and it works quite well in high dimensional situations. Once we've got the clustering, we can then look at variable importance. And here you can see some results from that. And this is basically slightly complicated because it's a Python script, and Python starts its numbering system from 0. So what most people would call clusters 1, 2, and 3, Python calls 0, 1, and 2. But essentially this tells you for feature one, which is this is the variable that's most important for clustering class 2, which in fact is cluster 3 out of the 4 clusters here. Whereas, you know, some of the other features are more important in clustering other clusters. So this gives you an idea of which features, and features by this I just mean variables, which variables are driving which clusters. This particular method, the SHAP method, is one of the most fashionable or state-of-the-art methods that's around at the moment. One of the other things we can do is multi-view clustering. Multi-view clustering is when we might want to cluster, say for example, genetic data plus imaging data, and look for similar, basically groups of individuals that cluster similarly across those two different sets of data, modalities of data. We can also look at how likely it is that a variable and an individual belong to a particular cluster. And here, the likelihood of a subject being a member of a cluster is shown by the size of the data points. So you can see that where the clusters get very close together, the points between the clusters tend to be smaller. And that shows that these points are actually switching between the clusters. Now, because this is not a Gaussian method, the way in which we compute the clustering probability of a subject belonging to a particular cluster is we just use all these many clusters that we've got. And here you see a very simple example. We've got 2 clusters and we've clustered them 5 times, going from left to right. The top cluster is basically the orange ones and the bottom one is the blue ones. You can see that 9 out of the 10 subjects in those 2 clusters of 5 are clustering identically across all those clusterings. So they've got a 5 out of 5 probability of belonging to their respective clusters, but point X clusters 3 times in the top cluster and twice in the bottom cluster. So that's got a 3/5 probability of belonging to the orange cluster and a 2/5 probability of belonging to the blue cluster. One of the other things we could do is a heat map type approach. We can actually take the data and we can actually bootstrap subjects. We can bootstrap resample the subjects and just recluster the data lots of times at the best cluster number that we've got. From that, we can get all sorts of information about cluster stability, i.e. the number of times that 2 subjects will occur in the same cluster, which is the sort of going from dark blue through to green and then through to yellow is the color of the sort of main panel on the left. But we can look at that in respect of, well, what does that mean in terms of variable values or group membership or whatever? So lots of information we can get out of that. And we've used that quite a lot in the past to try and illustrate lots of features of the clustering in a single slide. So basically, at the moment, the method I've just described is implemented in the R programming language, a script that we actually implement in RStudio. It requires a considerable number of R packages to run, including a modified form of the density algorithm to encompass all the changes that we've made. That algorithm is freely available to anybody who wants to use it. And basically, it seems to work extremely well on the data that we've used so far, both the test data sets and also give biologically plausible results on the data that we've been using in the study that Julia is going to talk about. Thank you very much for your attention.
So Mick has given you a great overview of the clustering pipeline. I was very happy to collaborate with him and be able to use it. And so what were the research questions which we were interested in answering? So firstly, I was interested in identifying neurocognitive markers and mechanisms that allow us to understand which autistic preschooler will develop ADHD features. Secondly, I was interested in whether cognitive ADHD profiles occur concurrently with behavioral manifestations of ADHD and whether they're associated with more behavioral clinical difficulties. And lastly, whether these distinct neurocognitive subgroups have unique neuroanatomical underpinnings. So this is just an illustration of the analytic strategy. I derived variables from 3 touchscreen measures that I've previously talked about and run them through a mixed density-based algorithm. I subsequently compared the subgroups on clinical and behavioral measures, which were parent reported, including the social responsiveness scale, ADHD rating scale, strengths and difficulties questionnaire, and sensory experiences questionnaire. And lastly, our study included some adaptations to the MRI protocol. So we were able to acquire MRI data in a sleep pediatric protocol. So I was then able to validate my subgroups on neuroanatomy. And what I found is a 4-cluster solution with 2 subgroups enriched by autism and with higher ADHD features, so subgroup 1 and 3. And referring to mixed indices of cluster probability, these subgroups had a very high probability of belonging to a given cluster of over 0.93. And subgroup 1, which was the executive functions and reward subgroup, as the name suggests, had specific difficulties in reward processing and executive functions and higher scores on the ADHD rating scale. However, we also found a low IQ cluster that had executive functions difficulties, often due to difficulties with task completion, and intermediate ADHD features. So you can see it in light blue. So subgroup that had neurocognitive difficulties across all domains had comparable IQ to other subgroups, but this intermediate subgroup showed general intellectual difficulties. So moving on to clinical and behavioral validation, we found that subgroups with elevated ADHD traits differed on all of the subscales of ADHD rating scale, so the inattention and hyperactivity impulsivity subscale. They also had higher SRS total score and higher scores on all subscales of the SRS, which is indicative of obviously higher autistic traits in children who have these neurocognitive differences. And whilst looking at strengths and difficulties questionnaire, we also found that these children have problems in prosocial behavior and peer relationship subscales. And the largest effect sizes were in the social communication SRS subscale, the inattention subscale on the ADHD rating scale, social cognition and prosocial behavior subscales. So we then proceeded to look at the underlying brain structure to better understand if any neuroanatomical differences are already present in preschool age and whether they could be contributing to these neurocognitive differences. So to derive indices of cortical thickness and surface area, we used something called FreeSurfer, which is a well-validated pipeline. And we derived cortical thickness, which is a measure that describes the distance between the innermost and outermost edges of the cerebral cortical gray matter, as well as surface area, which is the cortical area of the outer layer of the brain. So here are some of our structural MRI results. So clusters 1 and 3, the ones with neurocognitive difficulties and elevated ADHD traits, were compared to neurotypical clusters in terms of neuroanatomy. And the executive functions and reward subgroup had decreases in surface area in regions such as superior frontal and middle frontal gyrus or precentral gyrus, and increases in parietal, precuneus, and lateral orbitofrontal cortex. So the regions that were implicated were mostly in the frontoparietal cortices, whereas the low IQ subgroup differed in areas like lateral orbitofrontal cortex, postcentral gyrus, or fusiform gyrus. So we also wanted to dissociate the effect of autism, as subgroups both 1 and 3 had a higher proportion of neurodivergent children. However, the results remained significant after covering for the effect of autism. So what this suggests is that we are indeed tapping into more dimensional variability that could be transdiagnostic across autism and ADHD, rather than a categorical variability and the effect of autism only. When we compared autism and control in our sample on neuroanatomy, we didn't find such widespread anatomical differences at that age. What we find is obviously differences in regions previously implicated in attentional networks, impulse control, and in processing reward. However, obviously, it's the conjunction of different difficulties across all 3 domains that seems to be contributing to this neuroanatomical profile. So methodological advances allowed us to identify subgroups of children who show ADHD features at age 3 to 4. And what we learned is that autism and ADHD at that age could manifest with isolated difficulties in neurocognitive domains, or could be associated with low IQ. And autistic children with ADHD traits already show compounded difficulties in preschool age and might need additional support for these differences. And there are specific neuroanatomical correlates of these differences. So what I want to highlight is that we wouldn't be been able to conduct this research without a conjunction of methodological advances. So firstly, we derived obviously a composite phenotype from a scalable touchscreen tool. We were then able to utilize a large, well-characterized cohort of autistic preschoolers. And then with mixed support, we could use a subgrouping algorithm that was robust, that we had methodological kind of trust in. And overall, this research advances precision medicine approaches where we could look at individual patterns across multiple levels and integrate a child's behavior, cognition, and brain structure. So considering that neurocognitive development is quite nonlinear and dynamic during preschool years, we obviously want to assess the stability of our solution. So actually today is a data freeze for the time point 2 of the PIP study. So we'll be able to look at how these subgroups predict ADHD features at after 12 to 15 months. So I'm planning these analyses as soon as possible. And also I want to replicate the subgrouping analysis in a sample of a pure ADHD group to better understand how profiles within a pure ADHD at that age predict different clinical and behavioral features and how they map onto neuroanatomy. So I want to thank my supervisors, so Professor Eva Loth and Professor Christina Ecker, and the entire PIP team, all the PIs and RAs who were involved in testing. And obviously a huge thank you goes to all of our little preschoolers who we call brain explorers, who kindly gave their time to participate in PIP. Thank you so much for your attention, of course.